नमस्ते बिटिया, हमेशा खुश रहो बिटिया। I am Dr. Bharat Sharma, Assistant Professor, Department of Pathology, Faculty of Homeopathic Science, JYOTI Vidyapeeth Women's University, Jaipur. Today's topic is Immunology, Thymus, T cell development part 1. During initial stage of the fetus development, the hematopoietic cells develops into the yolk sac. After some times, they migrated to liver and then finally migrated to the bone marrow. Yolk sac, now primitive hematopoiesis goes to the fetal liver, then goes to the bone marrow, and this is the primitive hematopoiesis, and in some cases definitive hematopoiesis, this goes to AGM and goes to fetal liver, then goes to bone marrow. In newborn, most of the development of hematopoietic cells occur into the bone marrow. Now we are going to learn how is the development is of T cell occur bone marrow and how it is transferred to thymus for the maturation. First, we have a clear concept about these pluripotent stem cells, totipotent stem cells, and stem cells. This is a diagram showing adult stem cell is pluripotent stem cells, mesoderm, endoderm, and ectoderm, and they derive tissues like cardiac tissue, skeletal muscle tissue, tubule cells of kidney, RBCs, smooth muscles, lung cells, thyroid cells, pancreatic cells, and ectoderm derived skin cells, neurons, and pigment cells. Totipotent stem cells is totipotent stem cell convert into pluripotent stem cells like blastocytes, go to multipotent stem cells like trigeminal embryo and embryogenic tissues and adult specific tissues. Stem cells is first divided into two part and then one part is developed again revolve into stem cell and differentiation of the bulk occur after first cells. Now, pluripotent stem cell is divided into lymphoid progenitor cells and myeloid progenitor cells. Lymphoid progenitor cells give development to T cells, B cells, and natural killer cells, while myeloid progenitor cells give development to RBCs, platelets, granules, granulocytes like neutrophils, basophils, eosinophils, and monocytes also causes in monocytes present in inflammation comes out of ECS as macrophages and resident monocyte present in brain like microclear cell, lungs, alveolar monocytes, bone, osteoclast, liver, Kupffer cells, and kidney mesangial cells. Pluripotent stem cell, myeloid progenitor cell, and lymphoid progenitor cells come in blood circulation, then attracts toward thymus by thymotoxin substance, reach in thymus. This is thyroid gland, this is trachea, this is the thymus, it is the right lobe of thymus, it is the left lobe of thymus, there are septae and lobules. This is the inner structure of the sinus, thymus. It is divided in thymocytes. It is trabecula. It is capsule. It is dead cells. These are thymocytes. These are nursing cells. These are cortical epithelial cells. There are blood vessels, Hazel's corpuscles, and interdigitating dendritic cells. This is epithelial cells microscopic diagram. Here is you can see the too much neutrophils. There is a diagram showing blood-thymus barrier developing T cells. It is epithelio reticular cells. These are basal lamina. This is the pericytes and this is perivascular CT. Beginning with begins with arrival of a small number of lymphoid precursor migrating cells from blood to thymus. When they do arrive in thymus, T cell precursors does not express signature surface markers like CD3, CD4, and CD8. Double negative do not express RAG1 and RAG2 that are necessary for gene rearrangement. Genetic changes occur in the cell which causes the cell to form a special type of protein which after formation travels up to the surface of the cell and denotes as the receptor, and these receptors are called TCR receptors. Positive selection result in MHC restriction mechanism. Immature thymocytes cluster with MHC molecule on the cortical cells of the thymus. If TCR interact with MHC, protective signal results that prevents apoptosis. If TCR does not interact with MHC molecule, no protective signal and apoptosis occur. Results only reactive thymocytes survive. Now selection process in thymus. Positive selection, survival of only T cell whose TCR recognize self-MHC molecule. Negative selection eliminates T cells that react too strongly with self-MHC or MHC with self-peptides. This is a diagram showing T cell receptor positive and negative selection. Negative selection ensures self-tolerance, weeds out high affinity thymocytes. Mechanism: APCs bearing MHC interact with thymocyte. If avidity is too strong, thymocyte undergoes apoptosis and result only self-tolerant thymocytes survives. T cell development is expensive for host. 98% of all thymocytes do not mature, dies by apoptosis within thymus. T cell differentiation. CD4 plus and CD8 plus cells leaves thymus and entire circulation in Z0 phase. Naive cells condense chromatin like little cytoplasm, about twice as many CD4 plus. Naive cell recognize MHC antigen complex, initiate primary response after 48 hours, enlarges into blast cell and undergoes repeated rounds of cell division, differentiated into effector cells, cytokine secretion, or B cell help, or memory cells, long lived response with heightened activity, secondary response. This session is powered by Digital Version 2.0, Jyoti Vidyapeeth Women's University. I hope you all enjoy this session. If you have any query, please mention it in comment box. Thank you.